So much of the microscopy that we're doing nowadays is fluorescence microscopy, and I'll be explaining what that is in this lecture. So why do we use fluorescence in the microscope? And as you have probably seen so far, there are two important aspects in making a great picture in your microscope. One is resolution. You want a high resolution. And the second thing is contrast. You want high contrast. Otherwise you wouldn't see anything. And what fluorescence offers is this high contrast. So it's demonstrated in this image here. We have part of the cell labeled in green. In green we're looking at microtubules. So we labeled microtubules with a fluorescence dye. And we have part of the cell labeled in red, and that part is actually the nucleus, it was DNA. It's a red label attached to the histone protein. And what you see here is that this label stands out on a black background. So we have a very high contrast. We see what we want to look at, whereas our background is totally absent. So it gives an actually extremely high contrast. Then what we also already see in this specific image is that you get specificity. We can attach these fluorescent labels to specific proteins, to specific molecules, to specific dyes, to specific places in the cell or wherever we want them. So we can target them that they show what we want to see. Then fluorescence is quantitative. So if I calibrate my system correctly, areas where we have higher signal, like here, those are areas where I have more of the protein that I'm interested in. So in this case, there's a lot more tubulin here in these bright areas than here in these dimmer areas. And then lastly, and very importantly, fluorescence is compatible with imaging of living cells. So this image here is not only a still image, but it's actually part of a sequence of images that I took. And when we now start playing those faster, we see that it's actually a dividing cell. So the microtubules assemble in this spindle, and that spindle then pulls apart the chromosomes into two daughter nuclei. So fluorescence is a great thing for microscopy, but what is it really? And what's kind of surprising to me still is that humankind has been aware of fluorescence for only about 150 years. So one of the first descriptions of fluorescence was by Sir William Herschel. He was studying the aspects of an extract of the bark of the cinchona tree, and he held up this extract in the light of the sun, and that came through his window, and he noted that the light of the sun induced a celestial blue light coming out of that extract of the bark of the cinchona tree. And I will now try to repeat that 150-year-old experiment here in the lab and be showing you that now. So the bark of the cinchona tree is hard to come by, but luckily the active ingredient can be found in every supermarket in the form of contained in tonic water. And so I'll take this tonic water, pour it in a glass, and then the other part is that we need UV light to illuminate it, and that you can buy in the form of what's called a urine tracker in the pet store. And so when we now shine this UV light in the quinine, in the tonic water, and you see that you get a blue light emanating from within that tonic water. It's another wavelength. It's a longer wavelength than this light with which I illuminate. And that's why we see it a lot better. Our eyes are not very sensitive to this UV, but they are highly sensitive to the blue light in the tonic water. Quinine is not the only compound that fluoresces, of course, otherwise it will be quite uninteresting. Already in the 19th century, scientists synthesized this dye called fluorescein that is highly fluorescent. And when I shine blue light in that, we get yellow fluorescence coming out. And another very well-known dye is rhodamine. And when you shine green light in that, you get this orangey, yellowish fluorescence coming out. And so this is fluorescence. Okay, so what we have seen is that we use light of a lower wavelength, so in this case of the quinine from the extract of the bark of the cinchona tree, we use UV light, and then the dye responds to that light by emitting light of higher wavelength, so in this case blue light. So in general, except for very special cases, emission light is of a longer wavelength, and that means lower energy, so there's some energy loss going on in this process, than the excitation light. Now this aspect of fluorescence is very important, and we often show that by measuring the spectra. So we measure an excitation spectrum, which is often very similar to an absorption spectrum, and we measure an emission spectrum. So here, that's this curve here, which is the fluorescent light emitted by the dye. So we characterize these dyes by their excitation maximum and by their emission maximum. Those numbers tell you immediately a lot about how to use that dye in your actual experiments. The difference between the excitation and emission maximum is called the Stokes shift, after Sir Stokes, who first realized that there was this difference. And you can have dyes that have a small Stokes shift, where the emission is very close to the excitation, or you have dyes with a large Stokes shift, where the emission is very far away from the excitation light. Now, so there's energy loss going on, and that causes this difference in wavelength. But how does that happen? And one of the most useful ways of thinking about that is through these so-called Jablonski diagrams. Jablonski was a Polish scientist who did this work at the beginning of the 20th century. And in these Jablonski diagrams, we use we're looking at the orbitals that electrons can be in. When an electron when a dye absorbs light, that electron is going to be kicked in a higher orbital, in a higher energy state. This absorption goes very, very fast. So it happens at the time scale of femtoseconds. And femtoseconds to me don't mean much, so I like to express this in the distance that light travels during that time period. And since light goes extremely fast, to me it's pretty amazing that light travels only like 0.3 microns during this time period. And once that electron finds itself into these higher orbitals, through a process called internal conversion, it quickly falls back to kind of what you could call the ground level of the excited state. And that internal conversion is a tiny little bit of heat that dissipates this way, so we have a bit of energy loss here. And that happens on the level of time scales of picoseconds. And light would travel only 0.3 millimeters during that time period. Now, then the electron can hang out here for what is, in comparison to these previous time scales, almost eternity. Something on the order of many nanoseconds. And then it will fall back to the real ground state and emit a photon. And that photon will be of a wavelength less than the wavelength that was, of an energy that is less than the energy that was put in initially. And that means that that photon that's coming out is going to be of a higher wavelength. Now, not only this return to the ground state is not the only process that can take place when a dye is in the excited state. It's also possible that the energy is transferred to other molecules, or even it can return to, the electron can fall back to the ground state but not emit a photon. So there's also non-radiative decay. So we have a total number of events, some of which result in light coming out, some of which that don't. And we express the ratio of those two as the quantum efficiency. And the quantum efficiency is a very important parameter of the dye. It not only depends on the dye itself, it also depends on its environment. For instance, what other molecules are around the dye. But nevertheless, quantum efficiency is something you want to know and that you want to measure, or that you want to be told what it is. Another important aspect is the brightness. And the brightness, in the end, is determined by how well a dye absorbs the excitation light and what fraction of the excited state returns to the ground state under emission of fluorescence. So it's determined by both the absorption coefficient and the quantum efficiency. Now, another important aspect here is what we already talked about. How long does it take between absorbing a dye and for the dye to return to the ground state? And that is what we call the lifetime. And so it's mainly determined by how long it takes before we return to the ground state out of the excited state. So usually this is on the order of nanoseconds. Nevertheless, it's also a parameter that is dye specific, that is also dependent on the environment of the dyes. And there's a whole form of fluorescence microscopy called lifetime microscopy that actually reads out lifetime instead of only reading out the fluorescence itself. Nevertheless, what we're now going to talk about is microscopes that discriminate between the excitation light and the emission light. So how do we do that in a microscope? Well, the main thing is that we use filters. So we use filters that filter the exciting light so that we get out of our light source only the light that is of the wavelength that will excite our dye. That light will bounce off the dichroic mirror and then excite our sample after it went through the objective. The sample will start fluorescing, and that fluorescence then travels back through the same objective. It will now pass through the dichroic mirror, and then there's an emission filter that will let through the fluorescence, but that will completely block any scattered excitation light so that we will only see the fluorescence on our detector, which I symbolized here with an eye, but that could also be a CCD camera or whatnot. And so this is the simple, basic principle of a fluorescent microscope. So the most important aspect here, and the thing that makes it different from a normal microscope, is filters. And these filters need to be very good because they really need to discriminate this excitation light from the emitted light. So what options do we have there? Well, you can have filters that are based on absorption, that absorb certain wavelengths of light. Those are not that great in practice because their spectra are very broad, so they will also absorb a bit of the emission light if they're so they will not exactly do what we want them to do. And the type of filters that we mainly use these days are interference filters. So what are those? Interference filters are basically thin alternating layers that have different refractive index, so that we get interfaces there that can reflect the light. So you can almost think about them as thin, transparent layers that are about the wavelength of the light, or half the wavelength of the light, with semi-reflective coatings in between. So what now happens when light hits this filter is that part of it will be reflected, part of it will move through. The light that reflects will interfere with itself, and when the wavelength is such that it matches the wavelength of the light, you will get constructive interference, and that constructive interference will then in effect let the light pass through this filter. Whereas when the wavelengths are different from the two, the distance between the layers, we get destructive interference and it will be reflected out. Now the filter manufacturers can completely compute how these layers will behave, so they can design in the computer what wavelengths this filter should pass through and what wavelengths should be reflected. And you can basically order any type of filter that you may possibly need. Now, just to make this a little bit more clear, there's one example of interference that you're highly aware of and that every one of us have seen, and that is what you see in soap bubbles. So in soap bubbles, when light hits that, you will see reflection of all kinds of colors of light. The soap bubble itself has a thickness that is on the order of half the wavelength of a lot of the visible light. And you will get the same interference effect where some of the colors will pass through and other colors will be reflected, and that gives these beautiful colored patterns. Now we will break and show you some of these filters, and we'll also show you now here how these filters are assembled in a filter cube. Okay, so I have an example here of such an interference filter. This one is called a HQ47040, and the 470 is the center wavelength of this filter, and the 40 means the bandwidth. So this one will pass blue light, and so if I have my blue LED and I hold that up, I see the blue light coming through, and you will see that this LED indeed passes it through. Now when I would have a green light source and hold that up and go through this filter, you will see that it gets a lot dimmer. And not only that, you may also notice that it's actually a little bit of blue coming through, and that is because this green LED is not purely green but also has a slight bit of blue light in it. Then, the next part is this dichroic mirror. So this one is designed, again, such that it reflects blue light, so you will now not see coming anything through, but if I reflect it to you, you may be able to see that. And then the last part is this emission filter that will transmit the green light and reject the blue light. So in effect, when we would now place these three in this configuration in the microscope, illuminate from this side, go up, we have the objective and the sample here, come back through and go through this emission filter. That's the principle of a fluorescence microscope. These filters, this is exactly the way they are arranged in these cubes that were designed by Ploem, where we have an excitation filter, a dichroic mirror, and then an emission filter here. And so these will, as expected, reflect, and I'll now turn it to you so that you can see it. So the exciting blue light will be reflected to the specimen. The specimen will fluoresce green, and that will make it to the eyepiece or to the camera sensor. So we've seen now these filter cubes. There's an excitation filter, there's a dichroic mirror, and there's an emission filter. What's very important to know, and the information you should always have about these filters, is their spectra. So what are the wavelengths that are being transmitted and what are the wavelengths that are being reflected. And those spectra then will look something like as follows. So we have here an excitation filter that passes through this band pass, these wavelengths, these colors of light. The dichroic mirror is made such that it will reflect everything that is lower than a certain cutoff band and pass through everything that is higher, and then the emission filter will pass through the higher wavelengths but absolutely reject the excitation light. So when you now assemble a fluorescent microscope, your task will be to select filters that match well the dyes that you'll be working with. And what you will be doing in that case is to try to overlay the spectrum of the fluorophores with the spectrum of your filters. So I have here a spectrum of a dye that is probably Psi3, the cyanin dye. It excites in the green area, so around 550, and it has an emission maximum around 600. So if we now use a bandpass filter centered around 650 nanometers, that will easily reject the excitation light because it's far away from that. But you also note that the integral of it, that it only passes through a fraction of the emitted fluorescence. And that means that you're losing something like 75% or so of the emitted fluorescence. And in general, you really want to get all the photons that you can. So it's not a perfect situation. So if you now switch to a bandpass filter that is slightly lower, that is around centered around 610 nanometers, then you're catching a much larger fraction, like 75 or 80% of the emitted photons, and you'll be much happier because your image will be much brighter. Now, we'll switch to the lab and show how we're using these filter cubes to collect actual images. Here we have a real fluorescent microscope. So we have a light source back here. This is a mercury arc lamp. There's more in the tip on light sources about that. That light then passes through the main body and then hits here the dichroic mirror. So, and I'll show you now how this works. So this is basically a carousel that you can take out. And this carousel contains, or turret, contains these same filter cubes that we have been looking at in the lecture. So this one is sitting here. The light hits the excitation filter. I can take these cubes out like this. So the light hits the excitation filter. We're sitting in the microscope like this, goes through the excitation filter, hits the mirror, goes up, the fluorescence light comes back, makes its way through the mirror, and then here at the bottom we have the emission filter that goes down and in the end to the camera or the eyepiece. And now, since this thing is motorized, once it's sitting in the microscope, it can, with a push of the button, put in another of these filter cubes. And these cubes have been selected to visualize the dyes that we're interested in. And so usually there's one for things like DAPI, for things like GFP or fluorescein, and a cube for something like M-cherry or rhodamine. Okay? So when I now put this back into the microscope, and then open the shutter here in the back that blocks the light at the moment, we get blue light coming out of the objective. Then when with a push of the button, I put now another dichroic in place. Here we have green light for rhodamine or M-cherry. And if I go on, we get like red light for something like a dye like Psi5. And so just by switching these, we can select the wavelength of the light for excitation and the filter used for emission, and select the fluorophore that we want to image. So we've now seen how we can use the fluorescent microscope to collect individual channels, images of individual fluorophores. Then what we can do in the computer is put all those images together, give them a nice color, and end up with things like this. So here we have a staining of mitochondria in red, of DNA in blue, and what looks like actin stress fibers here in green. This works very, very well. There are also alternative strategies to doing this. And one is, what you may have noticed in the lab part, is that it actually takes quite a bit of time to move that turret. So going from one position to the other takes, quickly, like half a second or so. The turret is pretty big, so it has a lot of vibrations. If you want to go faster, the whole thing starts moving more. Those are all bad things and things you don't really want. So instead of using these filter turrets, what you can do is put your excitation and possibly also your emission filters on a filter wheel. Now I brought such a filter wheel and can show that to you here. So it really is a wheel that has individual filters in them. There's a motor that can, through this rubber band here, moves that around, and we can by activating the motor, we can switch between these different filters, and we can do that at a speed that generally is much higher than you can do on a turret. So we then first take an image where we put the first excitation filter in place. So for instance, in this case, we have a blue excitation filter, we get blue light out, we get green fluorescence. Then when we move the filter wheel to the next position, which has a yellow filter, we get red fluorescence coming out. So we collect now the second image, and that goes much, much faster. Now you may have noticed that there is something funny going on, because this dichroic filter here apparently reflects both the blue light and the yellow light, but it passed through the green light. And so that means that that dichroic filter needs to have pretty special properties and is very different from what I've shown you so far. And indeed, that is the case, and luckily the filter manufacturers are able to make such interference filters, so they can make filters that reflect at certain wavelengths and then pass through the light in between those wavelengths. And some of the spectra of filters you can buy look a lot better than what I'm showing you here. It's literally amazing what the filter manufacturers have accomplished, and that has been also a great driving force behind the whole progress in fluorescence microscopy. So we had our image of our red dye, and then we put that all back together in the computer in these multicolored images. Now, also that emission filter, we could, instead of having an emission filter, which would have to be a multi-band pass emission filter, we could replace that by a second filter wheel, and then we have full control and very fast control over both the excitation and emission filters. One practical aspect that you need to be aware of is PhotoBleaching. So fluorescent dyes don't last forever. They only have so many cycles that they can go through, and over time their fluorescence tends to bleach out. So for instance, in this movie that I'm showing you here, where I coupled GFP to the microtubule tip tracking protein EB1, we saw that at the beginning we had bright fluorescence, and then over time it completely disappears. Now this is regretfully a common aspect of most dyes, and it is something that we usually really don't like and that we want to avoid. So what can you do about it? First of all, you can select dyes that are resistant against fade or that do not bleach as fast as others. So for instance, this dye fluorescene that I showed to you before tends to bleach extremely fast. There are now other dyes on the market that have the same excitation and emission properties, but that bleach much slower. So you would almost never want to use fluorescene anymore, but use one of these replacements. You can try to label more densely. So each dye has a certain chance that it will photo bleach. So if we now put a lot more labels there, it will take much longer before all my signal is gone. Then, very importantly, you want to change the environment of the dye so that bleaching decreases. So for instance, oxygen is important in this photo bleaching process. So a common strategy is to remove molecular oxygen from your sample as much as possible. For instance, by using glycerol, you're already doing a lot better. Or by using enzymatic systems that take out the oxygen from the sample. There are also a lot of compounds that you can add to your mounting medium that will reduce bleaching. And lastly, and very importantly, you really want to use all the photons that you can get out of your dye. So you don't want to leave on that excitation lamp when you're not looking, when you're not observing your sample. So always close the shutter immediately when you're not looking. Or when you're doing camera-based imaging, you want to take the shortest exposure times possible using the lowest amount of excitation light that you can. And you also want to synchronize your camera with the shutter as tightly as possible so that there's no dead time in which you're not actually collecting that fluorescent light. Now, I'd like to finish this presentation going back to that movie that we saw in the beginning. In the end, we're using fluorescence simply because it's a great tool to let us look at cells and let us look at individual aspects of cells. And to me, it's still just amazing that we can see these living cells and components in these living cells while they're living. And the beauty of these kind of movies and images is just utterly stunning, and it makes it a joy to actually work with fluorescent microscopes.